Good afternoon. As people are coming in, we'll start on time to not shorten the available time for this lecture. As many of you know, this is the Florence Mahoney Memorial Lecture as part of the Walls of Wednesday Afternoon series. And I'll just remind people very briefly that this was the Florence Mahoney Lecture until she passed away at the age of 103 a few years ago, and then it became the Memorial Lecture. It honors her because she was really an incredible character and advocate for NIH and NIH research. Her interactions with numbers of former presidents and influential individuals in her famous Georgetown townhouse. For those of you who know the area, this is a very famous landmark, and the pictures of her interacting with many of former influential individuals led to the formation, in fact, of NIA as an institute. She served on councils of multiple institutes, and when I would meet with her prior to these lectures or prior to the determination of the speakers, she was always very adamant that she wanted serious science. She wanted to know what it was all about, and she'd be just thrilled with today's speaker, Tom Rando. Tom, by way of introduction, was born in Brooklyn, like it seems most of us were. Did his undergraduate MD and PhD work at Harvard, a year at MGH, an internship, and then moved to the West Coast, where he did his neurology training at UCSF, and then moved to Stanford, where he's been since that time. Now is professor and directing the Glenn program in biology of aging research there. He has been the recipient of multiple awards and has had an enormously prestigious career in pursuing mechanisms of aging, as the title of his work indicates, particularly looking at abilities to modify or reset the aging clock. And particularly, as you'll see, he has elucidated so much about the non-cell autonomous aspects of cell aging, the ability to influence the way in which cells behave by regulating their environment. He's applied unique techniques, not new, but newly applied, as he'll be describing to us. And so without taking any more from the hour we have with him, it's a great pleasure to introduce Tom Rando to you. Tom.
Well, thank you very much, Richard. It's a real honor to be here to deliver this lecture and actually to pay tribute to Florence Mahoney, about whom I've heard many wonderful stories, and her contribution to the research in the biology of aging and to the formation, in fact, of NIA. So again, thank you for coming. I'll be talking today about a general overview of our interests in the biology of aging with this provocative title of Resetting the Aging Clock. And I will give you sort of a general background and then talk about work we've done as well as where we're going in terms of our studies. So I often like to start with some other people's views of aging, and Shakespeare had a rather bleak view, I would say, of aging from this quote. I don't like to think of aging so much as rotting, but changing. But without a doubt, there is a function of decline that associates with what we think of as aging. And so what I'll talk about today are these general questions: is can aging be slowed? Can aging be arrested? And then finally, the main point is, can aging be reversed? With a thought process that really led us from one to the other to ask this, which sounds almost kind of a fanciful question of can aging be reversed? So this is a famous person, Jean Calment, who I think her story has stimulated a lot of interest in aging and how we age. She is the world record holder of the person who has the longest lifespan on record, 122 years. She died many years ago. I never had the pleasure of meeting her, but she had really quite a wonderful life story. And the question arises, as we look at people who are living older and older, why is it that some of us age more slowly and others age more rapidly? So I wanted to start off with a couple of general points, and the first point I want to make is that we often talk about aging and longevity in the same sentence, and I think this can be a problem because they're really quite different things. Aging is a process and longevity is a measure, and a lot of the advances in what we call aging research has stemmed from studies of lifespan. But in fact, aging and longevity can be dissociated because one can easily imagine that one can have death, which is the end of longevity, without aging. And then I'll mention briefly about the idea of aging without death. But why is longevity such an interesting endpoint? Is because, number one, it's very easy to measure and it's very reliable. So pretty much you can count on organism dying, okay? So that hasn't been a problem, and because it's so easy, people have used that as an endpoint of studying aging. And so it really has been a productive approach to thinking about how long an organism lives in terms of its relationship to how it ages. But this other flip side is, you can imagine death before aging, but what about aging without death? Well, this is just a mythical representation of what that might mean, but one can perhaps extrapolate that into the real world. So this is a picture of Tithonus, shown here, who had the actual misfortune of getting involved with the gods. He fell in love with the god Eos. And in a dispute with Zeus, Eos asked her to give her one wish, to keep Tithonus as her lover, and she asked that Zeus grant Tithonus immortality, which he did. But he didn't grant him eternal youth. So Tithonus lived forever, but he continued to age and age and age, and depending on how you read the story, he turned ultimately into this little cricket in the corner, chirping away and begging for death. So that's aging without death, and it doesn't happen in nature, but one can imagine lifespan as being an inaccurate measure of how we age. So can aging be slowed is the first question, and of course there's ample evidence from the experimental literature that it can, and probably the best example of this that many people will be familiar with is the idea of caloric restriction. And caloric restriction is really reduction in caloric intake, reduction in calories, while maintaining adequate nutrition. And this has been shown to extend lifespan basically in every species that's been looked at, from yeast to flies to worms to mice. This is one of the early studies in mice from Rick Weindrich's lab, showing a life curve of wild-type mice shown here, and then mice of increasingly reduced calorie intake, showing they live longer to the point where the average lifespan of the control versus the fully restricted mice is extremely different, with a much longer lifespan than the control. So if one extrapolated this to humans, this would mean that a human could live to 150, 180 years by an equivalent level of caloric restriction, which has not yet been shown. And yet, of course, one can engage in caloric restriction on one's own. There's a caloric restriction society in which you can share your agony of caloric restriction with fellow members of the society. But it has yet to be proven whether, in fact, this is truly going to work in humans, although there's certainly circumstantial evidence that it might be the case. But there's something else in terms of the idea of slowing aging that I think is embedded in how we age as a population. And this is just a lifespan curve for humans from the US. For people born in the early 20th century, mid-20th century, where there was very little infant mortality, so most people survived into the ages of 50 or 60, at which point people start to die. And from these you can, of course, extrapolate what the mean lifespan is, and this is what the actuarial tables tell us about what the life expectancy is of any kind of cohort. And what's been interesting is if one looks at the mean lifespan across a century, say starting in 1900, the mean lifespan in developed countries was between 40 and 50. And in the space of just about 100 years, that's almost doubled. Okay, and so now the mean lifespan is getting up to be about over 80 in this country. So that's really an extraordinary advance. Now, a lot of that, of course, initially was due to the prevention of infant mortality. So a lot more people were reaching the age of 50 or 60 so they could live to older age. But something else has happened, and I think we really don't understand that, and that is the idea that people who are 80 now are much younger than people who were 80 a century ago. And we don't really have any way to quantify that, but I'll just show you one anecdotal piece of information. So here's Charles Darwin as an older man, as an adult, and then as an older man. And if you look at these images and you try to guess how old you think he is, you're probably thinking in terms of today's life expectancy. But in fact, this is his age in these pictures. So I think you would probably agree that for a 64-year-old person today, this person looks much, much older. And just by way of comparison, to present another iconic person from our own generation. Here's a person 45 years old and in her mid-60s. So of course this is just a single example, but there's no doubt that something has changed in how we live that leads us to be actually younger at an older chronological age, and it's something we still don't understand. So can aging be arrested? I think there's a lot of good evidence that this idea of extending lifespan by slowing the process by which we age exists, but can aging be arrested? And how do we even think about that? Well, one example of this that I think is very interesting to ponder is the idea of really stopping the aging process that occurs naturally in nature. So this is a C. elegans. This is a worm that goes through these early stages of larval development for a few days, and then as an adult lives for 2 to 3 weeks. So that's the natural lifespan, natural curve of the aging of C. elegans. But what happens in the case of so-called unfavorable environment, which say, for example, is a deprivation of nutrition, these worms can enter what's called a dour state and exist in this state for several months, so much, much longer than actually the full lifespan of the organism, after which they can come out of this state if given nutrition and then live from then on a normal life. So what is this really? It really is a disconnect between the passage of time and the changes that we call aging. So it really is, in a sense, an arresting of the aging process that occurs in response to an environmental stimulus. And there are many such examples of these so-called dowers or diapauses, often during development, but they occur and they can lead to a much more extended lifespan than would be seen in the absence of these interventions. I put this up just as another really dramatic example of a disconnect between the changes that naturally occur and the passage of time. In this case, this is a report that came out several years ago in which some date seeds that were found actually from an excavation that dated them to the 1st century BC. Okay, these dates were found in the excavation. They were presumed to have arisen from that time. They sat on a desk for several decades, and then someone had the idea, well, what would happen if we plant them? In fact, they planted them and they grew plants. So this is again a case of a remarkable preservation of biological activity in what I would consider an arrested state, so a disconnect between passage of time and changes that naturally occur. So with those kind of ideas, I'll turn to this question of can aging be reversed? Now, they didn't emanate from these questions, but they really emanated from our own studies of stem cells. And so I'm going to present to you some of our studies of aging, but really through the lens of stem cell biology. And we've been very interested in stem cells and how they function, how they age, and how they exist in this context of an aging organism for a variety of reasons, one of which I've illustrated here, and that is the two ways in which we think of cellular aging. On the one hand, cells that divide are considered to undergo what's called replicative aging. That is, they divide and divide, and they have a limit to the number of times they can divide, the well-known Hayflick limit. So cells will divide and age that way, but then at the same time, there are cells that are not dividing, say, for example, a postmitotic neuron in the brain that undergoes what's called chronological aging. So a neuron that's in the brain over the course of decades will in fact show changes that occur without any cell division. Now, stem cells, particularly stem cells in the somatic tissues, many of them exist in a quiescent state where they exhibit chronological aging, but then also in response to stimuli may undergo cell division. So stem cells may have, or somatic stem cells, in fact, may have two ways of aging, and we've been very interested in trying to dissect which of these is most important for the kind of changes that we see in the function of stem cells with age. So one of our main areas of interest is in the stem cells that exist in skeletal muscle. And this is an example of such a stem cell. So what I've illustrated here, here's a single multinucleated muscle fiber. The muscle cells are among the few true syncytia in the body, so many nuclei in one cytoplasm. And associated with this single muscle fiber is a single cell shown here, a so-called satellite cell, that represents the stem cell compartment of skeletal muscle. And these are rare cells. They constitute perhaps a few percent of the nuclear content of an adult muscle, and they exist primarily in the quiescent state for the duration of a person's life. So we think that the turnover of muscle is very, very low, and that in fact these cells may sit in a quiescent state in humans for decades. And yet, in response to a stimulus like an injury, they can be called upon as reserve cells to engage in remarkable tissue repair and regeneration. And I've illustrated that here. Here's a histological section of uninjured muscle. In response to a total necrotic injury shown here, there are a few of these stem cells that will begin activating. They will proliferate, they will amplify a lot, at which point they will differentiate and then fuse into these small multinucleated cells that we refer to as myotubes at this stage, shown here, these red structures, and then these will mature into mature myofibers over the course of several weeks. So this is a process that has a remarkable regenerative capacity. It is a marker of how these stem cells can function, and these stem cells also self-renew, so you can repeat this process over and over and over again, and muscle will continue to regenerate. So I just want to discuss one signaling pathway that we found to be important in this stem cell lineage, because it comes into play in terms of our studies of the biology of aging. Several years ago, we started to ask the question of these quiescent satellite cells, what controls their transition to an activated state, a proliferative state, and then a differentiated state? And one of the pathways that we found to be very important was the so-called Notch signaling pathway. We found that it's highly induced early on. It leads to the activation of these cells and their proliferation, but then that signaling pathway must be inhibited for these cells to go on to a more differentiated state. This is just an illustration of that signaling pathway. The Notch signaling pathway is a ligand receptor signaling pathway in which both ligand and receptor, shown here, are both transmembrane proteins. So this pathway is activated when a cell bearing a ligand, in which case I'll refer to delta as one of the ligands, interacts with a cell bearing one of the receptors. That leads to a cleavage of the Notch receptor, and this intracellular domain of the receptor actually translocates the nucleus and acts as a transcriptional activator. So this is the model that we've been working on in terms of just understanding stem cell biology. This ligand delta is upregulated very quickly in response to injury. It's upregulated within the niche in which the stem cell sits. That activates the Notch receptor on these cells and leads to their proliferative amplification. So what happens with age? So I show this picture as a way of thinking about the regenerative properties of tissues in young and older males. And this is Picasso and Son. That's a story for another day. But if you look at the tissues in this individual, in this young boy, he has remarkable regenerative capacity across all tissues. An injury will lead to a rapid repair of that tissue with almost no scar formation. By contrast, as we age, as one gets older, if one injures skin or muscle or bone, it repairs, but it's less effective, it's less rapid, and often it occurs with a prominent formation of scar. So there's something that definitely changes over time in terms of tissue regenerative capacity. And I've illustrated this here for skeletal muscle in the mouse. This is a young mouse, an old mouse, equivalent to, say, a 70-year-old human, and a very old mouse, equivalent perhaps to a 90 or 100-year-old human. What I'm showing here is the regenerative response at an intermediate stage where these myotubes are being formed after a total necrotic injury. So in the young animal, you see these nice regenerating myotubes, a little bit of connective tissue in between them. As the animals get older, and we do the same experiment, we see fewer of these myotubes, more connective tissue, to the point where at this very old age, there's very little regenerative myogenesis and a lot more scar formation. So it really illustrates this declining regenerative capacity that occurs in lots of tissues. So we began to ask many years ago, what is the basis of this? Why is it that stem cells are unable to repair tissues in the very old animals and in humans compared to very young animals and humans? And it turns out that it's not the simple answer. It's not that we run out of stem cells. So in most cases, in most tissues, there are plenty of stem cells that exist in an old tissue, but they just don't seem to function properly. If you actually count the number of cells, there may be a decline with age, but not nearly enough to account for this profound defect in regenerative capacity. So one of the first experiments we did was to ask, if we understand this Notch signaling pathway is important, if we look at this pathway in young or old animals, do we see a difference? So what we're illustrating here is a response to an injury. The injury is shown by this orange stain here in the adult, and we're looking in green at the upregulation of this Notch ligand. And you see, adjacent to this injury, these are the muscle fibers, and they're upregulating this Notch ligand, stimulating the stem cells that are associated with those fibers to activate and proliferate, to begin to migrate into this area to do repair. But if we look at an old animal, we see almost no upregulation of this Notch ligand delta. So the stem cells are there, but they're not getting the signal they need to activate. So if this is the model we have in the young, if we look at the old, what we see is there's a failure of upregulation of delta, and so fewer of the cells activate, and fewer of them produce these differentiated myotubes. So this really accounts for this declining regenerative potential. Well, so we asked, if that's the case, if this is really one of the prime defects, what would happen if we actually directly stimulated the Notch pathway, not relying on the upregulation of this endogenous ligand? So this is an experiment that we did with Irina Conboy when she was a postdoc in my lab almost a decade ago now. So this is an area of injury in a control animal in which we've just produced an injury, and there's a little bit of regenerative myogenesis going on here. And this is the old animal again, so there's a lot of fibrosis. But if instead we take an old animal, we create an injury, and we inject an activator of that notch pathway, we're able to get regeneration that looked just like an adult animal. So this is really highly effective and very robust regeneration. So this is probably a turning point experiment, because this said to us that we know the cells are there, and we know that if given the right signal, even in an old tissue, one can stimulate a stem cell in an old tissue to repair as well as a young stem cell. So there's nothing irreversible that's happened intrinsically in the stem cell that would make it unable to repair a tissue. So again, if this is the model of the effect of aging, if we directly activate the notch pathway, we can restore that youthful regeneration to an old tissue. But we also asked the question, of the cells that do activate in response to this impaired aging environment, do they activate normally? And we actually had evidence that they did not, because we began to look at the cells that emerged in the setting of injury to an old muscle, and that seemed to be not nearly the same kind of response that we see in the young animal, not only quantitatively, but also qualitatively. So we've created quite a few mice that are genetic lineage tracers of muscle stem cells. This is an inducible Cre recombinase knocked into the PAX7 locus generated by our colleague Charles Keller. And PAX7 is a gene that is expressed in adult muscle only in the adult muscle stem cells. So this is a way of turning on a gene only in adult cells. We can cross that with a reporter strain, in this case the Rosa locus, which expresses LAC-Z. So when we cross those, we administer tamoxifen. The stem cells in the muscle express beta-gal permanently at that point, and only those cells. And we have other markers as well, so we can follow the fate of muscle stem cells in response to environmental stimuli, in response to aging. And what we've found is that if we look at the phenotype, this is from a young or old, we see cells here that are X-gal positive, means they were derived from bona fide muscle stem cells. From the young, they express the myogenic markers, MyoD and PAX7, pretty much always. But in response to aging, or in this case, response to old serum in which they're grown, many of the cells begin to express this different morphology, so a flattened morphology. They lose these myogenic markers like MyoD and PAX7, and they appear to adopt a more fibrogenic fate. So these are true muscle stem cells that in response to aging or an aged environment lose their ability to retain the myogenic lineage and become more fibrogenic. And this is just quantification of that kind of phenomenon. We rarely see these non-myogenic progeny of muscle stem cells in young, but we see really quite a high percentage of cells from old animals that start off as muscle stem cells, but then become fibrogenic. So in addition to this problem with notch signaling, leading to an impairment of the activation, there's also something that's leading some of these cells to diverge and become more fibrogenic. So that clearly contributes to the fact that when old muscle is injured, there's less myogenesis, and perhaps because there's more fibrosis. So our studies have led us to ask the question, if there are these defects in notch signaling, if there are these changes in cell fate, can we begin to understand what's regulating those changes in cell fate? And can we begin to understand the signaling that's different in an old muscle stem cell compared to a young muscle stem cell? And these are some studies that were done with Andrew Brack when he was a postdoc in my lab. He was looking at these so-called top-gal mice, which just are reporter mice that report on the extent of Wnt signaling in a tissue. And what Andrew did, this is a whole mount of a whole muscle from the mouse. He produced an injury in a young animal or an old animal, and he found that unexpectedly, the injured area in an old animal had a dramatically increased amount of Wnt signaling. And so we weren't sure what this meant, but he was wondering if this Wnt signaling activity might relate to some of the phenotype we see in old muscle stem cells. And what he found was that if he added Wnt to old muscle stem cells, he could drive them toward a fibrogenic lineage. If he blocked Wnt signaling, he could maintain them in a myogenic lineage. And this is some in vivo data where he's again looking at regeneration in young versus old animals. Now you see the regenerating myotubes in this sort of orange color, and the fibrosis is in green. You see with aging, many fewer myotubes and a lot more fibrosis. But if he introduces into an injury at this time an inhibitor of Wnt signaling, he sees much better regeneration and much less fibrosis. So here we're looking at the fibrotic index, and that's markedly decreased by blocking Wnt signaling. So we had this expanded model that not only is there a block early on of this notch signaling pathway, but with aging there's an increase in Wnt signaling which acts on the quiescent stem cell and drives them toward this non-myogenic lineage. So clearly another way in which aged stem cells are behaving differently from adult stem cells. So obviously these are all very interesting phenotypes of aging, and we were very interested in trying to understand, so what is it about this aging environment? Can we begin to ask the question, what changes with age in the environment of a tissue that is instructing, as we think of this, the old stem cells to behave differently? And so we wanted to come up with a system in which we could artificially, experimentally modulate the entire environment of a tissue without actually injecting drugs or injecting compounds, but where the systemic environment of that tissue would be changed from a young to an old state. And we turned to this technique that's been around for many, many years, actually many, many decades, called parabiosis. And this is the first report of this kind of grafting of animals by Paul Bert in the mid-19th century. It was a medical thesis in Paris, in which he was doing a lot of grafting of body parts and different parts of animals from one to another, and he decided to try and graft actually two animals together. And what he found is that when he grafted two animals together, they developed a single shared circulatory system, so the tissues of one animal were exposed to the circulating environment of another animal. And this has been used over the years to study aspects of immunology, endocrinology, and we kind of resurrected this for the study of aging and stem cell biology. So this was a photograph that one of our collaborators, Saul Vieta, had picked out. He's a student in Tony Weiss-Corey's lab. This is a painting by Frida Kahlo, and I don't think she was familiar with the writings of Paul Bert, but what she's drawn here, painted is herself as two people. This was after her divorce from Diego Rivera, and this is the morning of Frida, and you can see her heart is open here and she's bleeding, but she's connected to the happier days of Frida when she was happy with Diego. And the idea here is that one part of her is influencing the other part of her. So this isn't what we modeled our experiments on, but we did follow on the work of Paul Bert to create these so-called parabionts. So what we do is we take two animals, we open the skin, suture them together. Over the course of several weeks, they develop these spontaneous vascular anastomoses, whereby they develop a single shared circulatory system. So the control experiments are what we call the so-called isochronic pairs, so a young animal connected to a young animal, or an old animal connected to an old animal. But of course, the interesting experiment is the so-called heterochronic pairs. We have a young animal connected to an old animal. So we create these parabionts. We let the parabiosis process develop over many weeks. We leave them this way for several months so that the tissues of one animal are now exposed to the circulating milieu of the other animal. And the question is, in this old animal, now exposed to this youthful milieu, if we injure the muscle of this old animal, what happens to its regenerative capacity? Are there influences of the young animal on the regenerative capacity of the old? And of course, vice versa, are there influences of the old animal on the regenerative capacity of the young? So here we're looking at regeneration, an intermediate stage of regeneration in the isochronic pairs. And what we find is that the young isochronic pairs regenerate just as well as young animals. Here's some early myotubes being formed. This is the uninjured area of muscle. But in the old isochronic pairs, here's the uninjured muscle, here's the area of injury. We see a little regeneration along the border and a lot of fibrosis. And those are the isochronic pairs. But if we take the heterochronic pairs and we injure the muscle of the old mouse, what we see is excellent regeneration, just like we see in young animals. So clearly, the exposure of this old tissue to a young circulating environment was profound enough to really reprogram, we think, these old stem cells to behave like young stem cells, much in the way we had seen with some of our earlier intervention experiments. And if we look at some of the molecular characteristics, here looking at delta expression, that notch ligand, it's high in the young, low in the old. If we look at the heterochronic parabionts, it's sort of intermediate. So definitely improvement in the old in terms of expression of delta, and a suppression, on the other hand, in the expression of delta in the young. And basically, the conclusion of a lot of these studies is that whenever we've looked, the old animal has benefited and the young animal has suffered from these heterochronic pairings. So this is looking at enhancement of the age phenotype, looking at age liver. We're looking at proliferation of hepatocytes by BRDU incorporation. It's very low in old animals or isochronic pairs, but it's markedly enhanced in the heterochronic pairs. If we look at neurogenesis, now we're looking at the brain in the neural stem cell compartment, again looking by BRDU incorporation. Neurogenesis is known to decline precipitously with age. We can enhance that neurogenesis shown here, these proliferating cells in the brain by heterochronic parabiosis. And just like with the muscle, when we look at the molecular characteristics, not only is there enhancement of proliferation, but the molecular characteristics of the age cells are more like those of young cells. Here we're looking at a BRIM-CBP alpha complex that had been shown from Nick Timchenko's lab, is absent in young animals, but is markedly increased in old animals, shown here in the isochronic pairs, but in the heterochronic pair in the old animal, it's reduced and it's increased in the young. So again, sort of a normalizing between young and old. So these heterochronic effects clearly are having bilateral effects. So what systemic factors may mediate these age-related changes in stem cell functionality? So this is a direction that we've really taken in earnest now, beginning to try and identify factors that change with age, that are affected by parabiosis, and that in fact influence stem cell function. So this is a project that was done again in collaboration with my colleague at Stanford, Tony Weiss-Corey, in which we're looking at neurogenesis. And as I mentioned, neurogenesis declines dramatically with age. These are proliferating neurostem cells in the old versus the young. But in the heterochronic pairs, it's increased compared to the old isochronic pairs. So what we did is we looked in the blood of young and old animals, and we looked in the blood of the parabionts, and we identified those factors that both change with the parabiosis and increase with age, and in fact found a relatively small number of proteins and cytokines that fit this kind of criteria of showing both a change with age and an effective parabiosis. So we focused on a couple of these. I'll talk about CCL11, which increases dramatically as a function of age. And we asked, what would happen if you expose a young animal to CCL11 alone as a marker of an aging cytokine? And we were doing this by using non-invasive imaging, looking at neurogenesis in a living mouse, shown here by just this luciferase signal, this light signal emanating from the stem cell compartment. If we administer a vehicle alone, there's no change from day 0 to day 4, but if we administer CCL11 directly into the ventricle, there's a suppression of neurogenesis within a few days, and that's quantified here. So we've looked at these battery of proteins that change with age and looked for ones that are enhancing neurogenesis in old animals or suppressing neurogenesis in young animals. And recently we've taken and this is again work from Tony Weiss-Corey's lab, of taking directly plasma. So instead of doing parabiosis, just taking plasma from young or old animals and doing transfusions, doing injections of plasma, up to 8 to 10 injections over the course of a couple of weeks, to ask if we can mimic the effects of parabiosis by direct injections of plasma. And so what they found is that, in fact, young plasma injected into old mice enhances neurogenesis and leads to not only an improvement of cellular function, but behavioral activities measured by a variety of behavioral assays. And this is just two groups of mice in the training period, one of which then receives old plasma, and what we're looking at is the number of mistakes they make in this water maze test. So if they received old plasma, they make this many mistakes, but if they receive young plasma, there's an enhancement of neurogenesis and a reduction in the number of these errors that occur in this behavioral test and a variety of other behavioral tests. So it seems as if we're beginning to be able to reduce the complexity of the parabiosis experiments to simpler but still complex in vivo experiments, and then now to be able to do this in vitro by administering to cells and to tissues young and old plasma and asking what components are there are either suppressing stem cell function as a function of age or enhancing it from the young milieu. So this leads really to the question of, is aging reversible? And this is a painting from the 16th century, Lucas Cranach the Elder, and he's painting the Fountain of Youth here. And it's a rather, again, sort of bleak view of aging, because there's this bleak landscape where older women, for some reason, are all being carted in and examined. They enter the fountain and they emerge as young women. They're led into this tent where they're closed and they're enjoying the fruits of youth, of love and life and laughter. So the question really is there anything to this that really is reversing the process of aging? And our question is, with this intervention of heterochronic parabiosis, are we really driving young cells to become old and old cells to become young? Or is it really something just much more complex, where we're changing a young cell or young tissue to be different, but not really old, in the same way an old tissue to be different, but not really young? So we're really asking, are we really working along this aging axis in these kinds of experiments? And is the age state really actively maintained and reversible, is really our question. Well, there have been several studies that come out over the years, subsequent years, that have really convinced me that we're onto something in terms of the aging state being maintained and reversible. And this is a figure from a paper published from a colleague at Stanford, Howard Chang, who was looking at skin aging. And what he found was, if you look at old skin compared to young skin, there's a very strong NF-kappa B signature in old skin So he created a mouse in which he could turn on conditionally an NF-kappa B inhibitor in an old animal. And he asked, well, what would happen if NF-kappa B is present in old skin and driving skin aging? Is it necessary that it's maintained to maintain the phenotype of old skin? And what would happen if we blocked it? And so what he's looked at here is a couple of markers, and this is the so-called senescence-associated beta-gal, a marker of senescent cells. And very high, this is in blue shown here, it's very high in old skin, but if he induces this NF-kappa B inhibitor, he can reverse, not prevent, but reverse the amount of senescence that he sees by this marker. Also a marker, P16, shown in brown here, is also reversed, both markers of skin aging and senescence. So this, again, suggested that he wasn't delaying aging, but he was taking old tissue and making it young again by these biochemical markers, and he showed also that the skin thickness was enhanced in the mice in which an NF-kappa B inhibitor had been induced. This is a figure from a paper from Peng Zhen's lab at the University of Michigan, which again is looking at this idea not of delaying aging, but reversing. So what she did was she administered rapamycin to old mice. Now rapamycin is being used in a lot of studies to show, or to test for, and has been shown to delay aging and extend lifespan. But this is a question, what if you take an old mouse, administer rapamycin for a short period, and then isolate the hematopoietic stem cells? Do they behave more like young stem cells or old stem cells? And they did a variety of different kinds of assays, including competitive reconstitution assays. And in all of these assays, the young is shown in white, the old is shown in black, and the rapamycin-treated old is shown in gray. In each case, there's a restoration or a reversal of this aging process toward a more young phenotype, as if this single drug for a short time can restore a youthful phenotype to an old stem cell population. So our hypothesis really has been that it's aging state is an epigenetic state that's driven by extrinsic and intrinsic stresses, is actively maintained, and it's stable, but it's reversible. And so a corollary of this is that the rejuvenation that we're seeing in these different paradigms occurs by cellular reprogramming. So I want to focus on this idea of cellular reprogramming in the idea of aging, and I want to discuss it in the parlance of something that I think is very, very familiar to everybody in this room, and that's induced pluripotency. So this remarkable story of induced pluripotent stem cells focuses on the idea that one can take a differentiated cell, undergo dedifferentiation by the introduction of a variety of factors to produce these pluripotent stem cells, which themselves can then redifferentiate to form differentiated tissues. So this has been obviously a huge and remarkable success in terms of understanding pluripotency and other aspects of cell biology. But I would argue that there's something else interesting going on here that has received much less attention, and that is, in these kinds of experiments, these cells are not only differentiated, but they're old. I mean, in general, a lot of these studies are done by taking a somatic cell from an adult animal. So they don't have to be old, but they're not youthful cells as in pluripotent cells. They're adult cells that when they are dedifferentiated, they're also rejuvenated, because these cells are cells in which I would say the aging clock has been reset. That is, these are cells that are now able to give rise to a germline that is able to give rise to members of the species. So these cells then have the capacity to give rise to an organism starting from essentially an age of 0. So, and then these cells can then age again and differentiate, and one could do this over and over again. So in this sense, IPS technology is about resetting not only a differentiation program, but the aging clock. And this is something that has been known since the time of therapeutic cloning started to be conceived and developed. The same thing happens in therapeutic cloning. So this is Dolly the sheep, famous success of mammalian cloning. So this Dolly was generated by an adult, therefore a cell with age, somatic cell, by somatic cell nuclear transfer, creating a sheep that could then give rise to more sheep. So this is clearly, again, the notion of an old cell becoming a young cell, and then that process of aging and rejuvenation occurring over and over again. And it's so firmly embedded in our understanding of biology that I don't think we even often think how common this is, that it's really not even a surprising finding. Because really, when you think of any fertilized egg, we think of a human fertilized egg, that occurs by the fusion of two cells that are perhaps decades old, in terms of a sperm and an egg, fusing together and forming a cell that has an age of 0. This cell starts the aging process over. So whatever that may be, by some selection process, some protection of the genome, clearly there's something that occurs with every fertilization that is the basis of the essence of metazoan life. So this is a cell that has whatever aging aspects were present in the sperm and the egg have been erased, and this cell now has, again, an age of 0. So this idea of resetting is clearly not only of interest, but it's of fundamental importance for the biology of life. So the interesting thing about thinking about these IPS technology, fertilization, nuclear transfer, is they all occur together. Dedifferentiation and rejuvenation occur as part of all these processes. And our question is, can we uncouple this? Can we take cells that are old and make them young without dedifferentiating them? And that's what we think we're doing in these experiments, where we're taking old muscle cells and making them young muscle cells. We're not making them pluripotent cells. We're not removing their differentiation marks, but we're asking, are we making them young cells? And in the same with the rapamycin treatment, the NF-kappa B inhibition. So the question is, what is rejuvenation without dedifferentiation? And how do we ask that question? And these are the fundamental questions that we're now pursuing in the lab is, can we begin to ask, how similar are young cells and rejuvenated cells? Are they the same? Are they different? Are we really driving what is otherwise an old cell to become young in terms of rejuvenation? And secondly, how do we define cellular age at a molecular level? And I think this gets down to the real fundamental question of how do we define age of a cell? We really do not have any molecular definition of cellular age, and it really will be only by doing that that we will know for sure if we're converting an old cell to a young cell. So I'll present just some of the recent studies just to end the afternoon in talking about how we're looking at this at the transcriptional level and at the chromatin level. So we've able to purify these muscle stem cells at a very high purity by flow cytometry, and we can look at their transcriptional profile across the whole genome. And what we find if we compare old cells to young cells in these unsupervised clustering heat maps is that, first of all, one thing that's interesting is they're remarkably similar. So it's not as if with age the transcriptional profile of a cell changes dramatically. There are some interesting differences, but they're remarkably similar. But if we look at cells from these heterochronic parabionts, the young and old cells from these parabionts are more similar to each other than they are to young or old cells. So this idea that we're actually converging as somewhere in between is supported by these transcriptional data. And if we look at just the average expression, again, across the whole genome of young cells, all the transcriptomes in the young cells versus the old cells, they're very highly correlated. Again, very similar with a very tight correlation. But if we look at the parabiont cells, old versus young, they're even tighter. So there's even more of a strict correlation between stem cells that have been isolated from heterochronic parabiont young and old animals than from just young and old animals. So this transcriptional profile looks as if it really is finding kind of the median between young and old, and these cells are remarkably similar. If we do principal component analysis, we also see this kind of phenomenon. So here we see these three groups of samples clustering together from the young and clustering from the old. So we think of this then as the aging axis in this principal component, where the young cells are positioned here, and it's along this axis that aging is happening. And if we then look at the heterochronic parabionts, they're displaced off this axis because of, we think, the effects of parabiosis itself. But they're displaced positioned really in the middle of this axis between young and old cells. So the young heterochronic parabionts and the old heterochronic parabionts really are falling right in the middle of this aging axis. And indeed, if we look at specific genes, we find those genes that are downregulated with age, well, almost all of them are rejuvenated, meaning they return more toward the youthful state. And genes that are upregulated with age, most of them are rejuvenated. That is, they're downregulated more towards the youthful state. So the transcriptional appearance of old cells that have been subject to heterochronic parabiosis really does look like a rejuvenation by this kind of criteria. And so we've obviously dug down into kind of these transcriptional differences, and I'm just going to mention this. I won't talk about the data in detail. But if we look at key differences, even though the transcriptomes of young and old cells are similar, there are some interesting differences. One of them, we see differences in Wnt target genes, which is very interesting in light of our finding of Wnt as promoting this kind of aging phenotype. And the second is in terms of chromatin remodeling factors, and I'll talk about that in terms of some of our chromatin studies. And our colleague, again, Tony Weiss-Corey, is looking at the same kind of transcriptional analysis in parabionts, looking at the brain from isochronic pairs and heterochronic pairs. And they found, again, some interesting differences that occur, some interesting patterns of gene expression that change in isochronic and heterochronic parabionts to create these kind of network diagrams and to look for what are the key nodes that are changing in parabiosis that suggest that old cells are becoming young and young cells are becoming old. And they've in fact been focusing in on a couple of these genes that are known to be very important in regulating synaptic plasticity with age that may be more of actually a feature of the differentiated neuron than the stem cell itself. So if we ask this question about what are we doing with these kinds of interventions such as heterochronic parabiosis, is it really moving along the aging axis versus just altering the phenotype of the cells? We were really beginning to favor the idea that we really are beginning to look at movement along the aging axis without changing the differentiation state of the cells. So just the last few minutes, I'll just end by talking some of our ideas of looking at the chromatin state of these cells, because in the end, we think that understanding the nature of the chromatin profile, the epigenetic profile, is going to define what is ultimately a young and old cell, and will define how a young cell can become an old cell and young cell, much in the way that epigenetic profiles have really defined the pluripotent state from the differentiated state. So we were looking at both histone modifications and DNA methylation patterns. I'll talk just briefly about some of our studies of histone modifications. And our question is there a histone code that defines cellular age? Will we be able to ultimately pull out and say, an old cell has this pattern of histone modifications distributed in this way across the genome that's different from a young cell? And can we find those histone patterns shifting between cells in these models? So this is just some ChlP-seg analysis looking at modifications of histones just in young stem cells first. So we're looking at two specific marks, trimethylation of lysine 4 of histone 3 and trimethylation of lysine 27 of histone 3. So these are conventionally used to mark genes that are active and actively transcribed versus genes that are repressed. And then interestingly, these bivalent domains are genes that express both of these markers and are often, at least in the pluripotent stem cell literature, considered to be poised, not active, but capable of being activated. So what's interesting is that in young stem cells, if we look just at the histone code, the vast majority of genes across the genome are marked only by K4 trimethylation. Very, very few cells are marked only by K27 trimethylation. So again, mostly active, very, very few are repressed, but still quite a few genes express the so-called bivalent domain. And we've been looking at these bivalent domains and what genes show them, and it's been very interesting because we look at the analysis of the pathways that are encoded by these genes. They are pathways that we would not have predicted to be occurring in muscle stem cells. And so as we begin to study these bivalent domains, this is a little bit of an aside, we're beginning to understand that perhaps in somatic stem cells, bivalent domains mean something different than they do in embryonic stem cells. But if we get back to this, some of the gene targets that we're interested in, if we look at two of our favorite pathways, so the notch target genes and Wnt target genes, what's particularly interesting in young muscle stem cells is the notch target genes are all marked by K4 trimethylation, but not by K27, so indicative of active gene transcription, whereas all of the Wnt target genes are bivalent. They have both K4 and K27 trimethylation. So we don't understand this yet, but it's an interesting signature that we would not have predicted from what we know about notch signaling and Wnt signaling in young stem cells. Okay, so then if we compare young stem cells to old stem cells, the histone patterns, these epigenetic patterns, are turning out to be very interesting and unexpected. So here we're looking at just this active mark, K4 trimethylation, looking at young stem cells in yellow and old stem cells in blue, just looking at the distribution of these marks about the transcription start sites of all the genes in the genome. What we see is that the enrichment for K4 trimethylation is higher in the young than the old, but what's really interesting is we look at this repressive mark, there's a lot of enrichment in the old and almost no enrichment, as I've mentioned, in the young. So the young stem cells appear to have a real dearth of enrichment of H3K27 trimethylation in genes across the genome, whereas with age, there's a marked enrichment of this repressive mark across the genome in the old stem cells. And this is just another way to look at this. So now we're just looking at transcription start site here, 5' and 3'. Young stem cells are highly enriched for this activating mark. Old stem cells are also enriched, but less so for this activating mark. But if you look at this repressive mark, it's almost completely absent across the genome at transcription start sites for genes in young stem cells, but highly enriched across the genome in old stem cells. So this is the kind of data that we're getting. We're doing ChlP-seg analysis and other transcriptional analysis, trying to look at pathways in genes to understand what might be the important patterns that are emerging, not only in these stem cells, but in other stem cells. So if we look at neural stem cells young and old, if we look at skin stem cells young and old, do we find that some of the things we're finding are shared between stem cells as a function of age, therefore, thereby trying to come up with what is truly an epigenetic signature of stem cell aging. And then obviously the next step would be to understand how these modifications are occurring, and are these modifications what we're controlling as we intervene with heterochronic parabiosis or these other rejuvenating interventions. Okay, so I just wanted to end by returning to the fountain of youth and ask this question, what are we doing here, and are we learning something about the nature of aging? And I think we are. I think we're studying something that actually occurs naturally. Our interventions are, of course, unnatural, but it does raise the question of how we think about the age state and how we think about it being altered in a way to make cells more youthful. We're not talking here about trying to make entire organisms more youthful, but for example, one can think of an application whereby in an old individual, if you could administer a treatment at a time of an injury to make those stem cells more youthful and more active during the phase of regeneration, not in any chronic way and not distributed throughout the body, but in a temporally and spatially controlled way, one can envision interventions that would enhance tissue repair, enhance functionality in older individuals by modulating the chromatin state and the transcriptional state of those stem cells. Okay, so I just want to end by acknowledging a lot of the people in the lab who did this work. It really is a work of many, many people. In particular, a lot of the work on the epigenetics is done by Tom Chung and Ling Lu, two postdocs in the lab who are doing a lot of this work, starting with the transcriptional profiling and moving on to the epigenetics, and helped by a lot of other students and research assistants in the lab. I wanted to acknowledge my former colleagues, Irina Conboy, Mike Conboy, and Andrew Brack, who really led us up to these studies in terms of understanding the biology of aging and these parabiotic studies and the signaling pathways that control them. Our colleagues at Stanford, Tony Weiss-Corey, were very interested in studying these systemic factors. We work on this together. He works mostly in the brain and looking at both the transcriptome and epigenome, but also the proteome, which is one of Tony's areas of expertise, to understand which proteins are changing in the systemic environment that are influencing stem cell function. And my colleagues Anne Brunet and Steve Artandi, who we have a program project through NIA to study the mechanisms of stem cell aging and different kinds of transcriptional changes that occur and what influences them, how that controls stem cell aging across different tissues. And finally, of course, I want to acknowledge the amazing support I've had from the NIH and the NIA. It's been great working with the NIA and my program officers and the people in the institution. It's been, I think, a great interaction, great relationship with us. We're very grateful for the foundation. We have support from the Glenn Foundation for Medical Research, which funds a lot of this work. We've had support from the American Federation for Aging Research, and the Department of Veterans Affairs has also contributed to some of these aging studies. So with that, I'll stop, and I'll be happy to take any questions. Thank you.
Thank you. So I ask people to come up to the microphones with questions, and maybe while doing so, let me ask one question, Tom, about your interpretation of some of the parabiotic experiments. One could look at experiments in which you reverse the age-related defects in muscle stem cells or neurons as indication that there are selected tissues that have suffered as a result of age intrinsically, and then there are others that have not, so that muscle stem cells, neuronal cells, in fact, were preserved, and its defects elsewhere, so that responsiveness to delta ligand is okay, but delta ligand is missing. So that interpretation, if there are selective losses and one can replace them, the alternative is that there are pervasive changes in all of these cells, and it's only the net effect and environment in a more complex sort of systems way that result in reversibility. Can you comment on the ability to distinguish those two?
Yes, so we haven't tried to distinguish those in a sense formally, but I would say that anecdotally, when we've looked at functionality of cells, young and old cells, I just don't think we've ever seen examples of cells that have preserved functionality, intrinsic functionality across the aging spectrum. I mean, I think that's the implication of your first. So that would be the first thing. We haven't found that. That doesn't mean it doesn't exist. And I think that what's a unifying theory of a lot of what we have found is that really it is a systemic milieu that is influencing tissues across the body, and they can be modified systemically and influence every tissue. So we also have not found any tissue that is not responsive to changes in the systemic milieu. So while formally we haven't excluded, I think most of our data supports the latter interpretation.
Well, glad to hear from you that there is some resetting switches in epigenetics for reversing the possibility. I am thinking at my age of 71 with 7 billion people, is there any way we could have a graceful exit? And so we heard the word apoptosis and all those things, but that happens to the leaves, not the whole tree. So could we find some switches that help us to preserve the resources of this planet so that the younger generation could enjoy it better?
So you asked me, have we found I missed a couple of key words there, I think. Have we found?
For the elderly people like us to disappear in a graceful way.
Oh, I see.
Ripening, not rotting.
Ripening, not rotting. I mean, again, I think the main focus of the work, at least from my point of view, in any kind of translational application to humans, is about health span. It's not about lifespan. It's not to make people live longer. It's to preserve functionality, to enhance, preservation of activity and graceful exit, as you say, in a way that we could do tissue by tissue rather than systemically. So I don't know if that addresses your question or your. Work harder. We'll try. Thank you.
Hi, Jess Curtis from NIA. I have a question about the heterochronic parabiosis experiments. You are citing a lot of differences found in the old population with regenerative potential in the aged animal, but did you do the opposite and look at the young animal and see if maybe the old milieu negatively affected the regenerative potential?
Yes, so I think I probably breezed by that in part because our initial interest and perhaps the most exciting is the idea of reversing aging, but without a doubt, this is, there's no free lunch. So every experiment we did in which we do, and we have done, with heterochronic parabiosis indicates that the old systemic milieu suppresses stem cell function in the young animal, and at every level. And so the one piece of data that I showed is that we found this one cytokine, CCL11, that increases with age. If we administer even that protein, let alone the whole heterochronic parabiotic paradigm to the young animal, it suppresses neural stem cell function. So in some ways, we started off by being very interested in rejuvenating factors, and then became actually more interested because it seemed to be so, and it may be dominant in what I would call aging factors. So I think that in fact the dominant influence is the aging influence, and that it's actually more difficult to reverse the aging phenotype than it is to promote the aging phenotype in the young. But absolutely, in every tissue we looked at, the influence of the old parabiont on the young was a suppressive one.
Okay, thank you.
Tom, maybe one further question about the impacts of parabiosis in either direction, and the likelihood that they'll be mediated by one or a small number of factors versus many. If I remember correctly, in one of the questions looking at brain structure function, and identifying a number of candidate molecules, you're able, with an antibody to one, to reproduce some of the systemic effects. So that's manipulating one factor, but if I remember, that was in the wrong direction. That was decreasing function. In regard to the comments you just made, is it therefore likely to be easier to find any one of many things that will make things worse, but will it be more challenging to find one or small number of factors that will make things better?
So I don't know. I mean, I would predict that's probably true. I don't know that. But I think what maybe end up being interesting is that why would one factor do this? Because when we first had these heterochronic parabiotic experiments, everyone was asking, have you found the factor? And my response was, there's no way this is going to be a factor. And yet we start to find factors. So the way I think of this is really an interactive network in which if we intervene in one area, we can disturb the network generally to create a more youthful or more aged phenotype. But the real question is perdurance. I mean, that is, and we don't really know that here, is if we create a more youthful phenotype in a stem cell, does it last? I mean, does it last? Have we created, in the same way an iPS cell is a permanent iPS cell? You can, I don't think we have that. And so I think what we're doing is we're tweaking the system, but we have not yet reached a perdurant and permanent youthfulness or age state. And I think, to your question, I think it will be more difficult to have a permanent youthful state than a permanent age state, however, even if we do it with one or a few factors. Yeah.
So consistent with mythology.
Yes, exactly.
One last question, and let me just say, after this, we'll have a chance to, for those who would like to speak with Tom further, at a reception afterwards, which, thanks to support by FAS, will include some refreshments, which, as you know, the government cannot provide. And this will be in the library out the corridor this way. So one last question?
Yeah, thanks very much for a very provocative and intriguing Wednesday afternoon. I was intrigued by the pictures of Darwin. Do you have any other evidence that people were aging faster in the 19th century than just that picture? I mean, if you gave him a shave, it wouldn't look that bad. And he also did have a chronic disease. I mean, it's rumored to be Chagas disease. It's hard to tell, but he probably had some kind of chronic disease that he encountered in his travels in the Amazon.
Yes, so maybe unfair to pick one example and one that's so, I think, pictorially illustrative. I think the best data are the actuarial tables. I mean, I think the insurance companies will tell you that your chances of dying at the age of 60 now are vastly lower than they were a century ago. And this is even true over the decades, that this is continuing to change. Now, you know, are we just treating diseases of aging differently, or are people actually reaching the age of 60 healthier and more fit than they were? And so I think that, you know, that's the preponderance of...
So I'm saying, how could that be?
I mean, so that's a good question.
We have 47,000 kinds of inimical chemicals in our bodies now that our grandparents never had.
Yeah, I mean, that's why I say it remains unexplained, but I think, you know, we're not only keeping people alive longer by preventing chronic disease. More and more people are living to old age healthier, healthier, not just, you know. So I think it's one of these mysteries of does it have to do somehow with how we are, in fact, you know, vaccinating and nutrition, all the things that change that might have actually been beneficial in infant mortality are actually having much longer effects on health. I'm just speculating, but that I don't think it's so outrageous to think that that's true. Mm-hmm. I mean, because clearly there are places in the world now where the average lifespan is 45. And that's, you know, and then if you look at those people, they're not living to, you know, very to 100. So there is infant mortality, but there's also earlier death in older age.
Maybe the NIH played an important role.
There you go. Thank you very much. We look forward to the reception for those of you who can make it.